Culture media. Introduction. Culture media can be defined as the nutrient preparation provided to a bacteria for their growth artificial condition. In a laboratory, the growth of bacteria on or in a culture media is called culture of bacteria. Culture media is used for growth of bacteria depends upon the type of bacteria to be cultured. Some bacteria required simple media, which contains some inorganic salts and some organic compounds, whereas others requires some special media which contains some special ingredients like eggs, blood, serum, meat extract, etc. Viruses are generally not cultured on artificial culture media. To culture viruses, living cells or tissues of humans or animals are used or egg inoculation is done. Uses of culture media: to identify the cause of infection from the clinical sample so that proper treatment can be given. To study the characteristics or properties of microorganisms. To prepare biological products like vaccines, toxoids, antigens, etc. For bacteriophage and bacteriocin susceptibility. To determine sensitivity to antibiotics. Classification of culture media. Culture media can be classified on various bases. Some common bases of classification of culture media discussed below. A. On the basis of composition. Natural media. Semi-synthetic media. Synthetic media. B. On the basis of physical state. Solid media. Semi-solid media. Liquid media. C. On the basis of use or utility purpose. Simple or basal media. Enriched media. Enrichment media. Selective media. Differential or indicator media. Transport media. Anaerobic media. A. On the basis of composition. 1. Natural media. Those media which are naturally present in environment and whose exact chemical composition is unknown are called natural media. For example, blood, milk, urine, meat extract. Two. Semi-synthetic media. Those media which are partially present in environment whose chemical composition is partially known are called semi-synthetic media. For example, Zapex media, sucrose, agar, etc. 3. Synthetic media. Those media which are artificially synthesized and whose exact chemical composition is known are called synthetic media. For example, Zapex solution, Richard's solution. B. On the basis of physical state. 1. Solid media. Those are solid in nature and contains 1 to 2% agar. They are usually prepared in Petri dishes but can be also prepared in test tubes according to need. Solid media are commonly used to study colony characters of bacteria and the change produced in the media differentiate one bacterium from another. For example, nutrient agar, McConkey agar. 2. Semi-solid media. Those media are semi-solid in nature which contains 0.2 to 0.5% agar. They are gel. They are commonly used for transport and motility test of bacteria. For example, Carey-Blair medium, amines transport medium, etc. Three. Liquid media. Those media are liquid in nature which do not contain agar. These media are generally called as broth. They are used for the development of bacteria and for some biochemical test. For example, selenite F broth, tetrathanate broth, nutrient broth, etc. On the basis of use or utility purpose, simple or basal media. These media are simple type of media which support to growth of those bacteria which do not have special nutrient requirements. These media are also called as general purpose media. These media are commonly used prepared enriched media to maintain stock culture for subculture of bacteria from selective media before performing staining and biochemical tests. For example, nutrient agar, nutrient broth, etc. To enriched media. These media contains some special substances such as blood, serum, eggs, so they are called as enriched media. When these special substances are added to basal or simple media becomes enriched media. These media are commonly used for growth of fastidious organisms. Fastidious organisms are those organisms which have some special nutrient requirements. For example, blood agar, chocolate agar, Loeffler's serum medium. Three. Enrichment media. They are broth media which are commonly used to increase the number of organisms in a sample. These media contains some specific substance which either helps to increase the number of a particular organisms or inhibits the growth of other bacteria. Particular enrichment media enhances the growth of particular organisms. For example, selenite F broth, tetrathanate broth, trypticase soya broth, Algae media, Loeffler's media, etc. Four. Selective media. These are the media which selectively enhances the growth of a particular bacteria or some particular bacteria but inhibits the growth of other bacteria. These media contains some specific substances which allows the growth of selective bacteria and inhibits the growth of other bacteria. For example, XLD, xylose lysine deoxycholate, and SS, Salmonella Shigella, agar, are used as selective media for Salmonella and Shigella. TCBS, thiosulfate citrate bile salt sucrose, agar is selective for Vibrio cholera. MSA, mannitol salt agar, is selective for Staphylococcus aureus. LJ, Lowenstein-Jensen, media is selective for Mycobacterium tuberculosis. 5. Differential or indicator media. These media are commonly used to differentiate one bacterium from another. These media contains some special indicator substances which produces changes in color of media after the growth of bacteria. According to the changes in the color, bacteria are differentiated from one another. For example, McConkey agar is a differential media which differentiate lactose fermenter bacteria from non-lactose fermenter bacteria. Lactose fermenter bacteria produces pink-colored fermentation colonies, whereas non-lactose fermenter produces pale, light yellow, colored colonies on this media. Six. Transport media. These media are commonly used to transport of bacteria or bacterial sample from one place to another place. These media helps to keep the bacteria alive during the travel period. For example, Carey-Blair medium, Stewart's medium, amines transport medium, etc. Seven. Anaerobic media. RCM, Robertson cooked meat, media is used as an anaerobic culture of bacteria. This media is prepared with meat extract from beef heart muscle. This beef heart muscle contains reducing substances and unsaturated fatty acid which absorbs the oxygen present in the media and make the condition anaerobic, which is suitable for the growth of anaerobic culture of bacteria.